Hello, I'm Jose Federico Lobato, and I'm going to present to you the poster called Nanostructured Biosensor for Detection and Quantification of HPV 16 and 18 with Plasmon Response. This is the work that me and my co-worker David Andres Camacho Morales are doing at the ITESO University. Well, first we have a brief introduction. Well, cancer arises from the transformation of normal cells into tumor cells in a multi-stage process that generally progresses from a pre-cancer lesion to a malignant tumor. These changes are the result of the interaction between a person's genetic factors and other external agents like physical, chemical, and biological carcinogens. Biological carcinogens are infections from viruses, bacteria, or parasites. Specifically, oncovirus, which are the one that we are attacking in this biosensor. These oncovirus infections promote tumor genesis utilizing metabolic pathways. The method by which oncovirus cause cancer varies by the oncovirus genetic material, DNA, RNA, or retroviruses. One common way is to induce uncontrolled cell growth by interfering with the tumor p53 gene. Human papillomavirus or HPV are, or however we're going to call it, is a non-enveloped double-stranded circular DNA virus that is responsible for causing multiple epithelial lesions and cancer. It can manifest as cutaneous and anogenital warts, which depending on the subtype may progress to cancer. The clinical lesions may be visible, but in some cases may require testing for viral DNA. Papillomaviruses are small. These viruses have an icosahedral DNA, which with a diameter of 52 to 55 nanometers. The bioparticles consist of a single double-stranded DNA molecule of about 8,000 base pairs that is bound to cellular histones and contained in a capsid composed of 72 pentameric capsomers. Once HPV is acquired, recurrences are common. For most patients with genital warts, there are treatments. In about 60% of the cases in women and men, genital warts resolve spontaneously. The biggest concern with genital warts is the risk of cervical cancer, but HPV is also known to be associated with anal, head, and neck cancers. The problematic is that in Mexico has a population of 49.6 million women ages 15 and older who are at risk of developing cervical cancer. Current estimates indicate that every year more than 9,000 women are diagnosed with cervical cancer and more than 4,000 die from the disease. This is approximately 45% mortality. Cervical cancer ranks as the second most frequent cancer among women in Mexico and the third most frequent cancer among women between 15 and 45 years of age. The first place is mammary cancer. About 4.1% of women in the general population are estimated to harbor cervical HPV 16 and 18 infection at a given time. And 65% of invasive cervical cancer are attributed to HPV 16 and 18. So obviously, the high risk and the cervical cancer is caused by HPV 16 and 18. And this is why we are going to focus on these HPVs. One of the biggest problematics is that the average female patient does not have access to a human papillomavirus test or has it for a low-risk HPV test. These low-risk HPV tests do not cover HPV 16 and 18. And a screening in Mexico for high-risk HPV and all the types of HPV that are possibly detected, because some are not possibly detected with some tests, cost around 3,500 or 5,000 pesos, which is $277 approximately. But the accessibility to this test is low, because when the Papa Nicolau is done in the women, some samples have to be sent to another country, because in Mexico, the clinics that do these tests are low or do not have the money to do it on site, and sometimes a lot of false readings are well, some tests are not reliable. Well, what we're using for the biosensor are two things: nanoparticles and the plasma response that the nanoparticles have, and the oligo and HPV L1 section. Well, first we have the nanoparticles, which are a wide class of material that include particulate substance, which have one dimension less than 100 nanometer at least. Depending on the shape of these materials, well, this can be 0D, 1D, 2D, or 3D dimensions. The nanoparticles showed characteristic colors and properties with a variation of shape and size, which these characteristics can be utilized in bioimaging applications or biosensing. The nanoparticles are conformed of three layers. First, the surface layer that can be functionalized with a variety of small molecules, metal ions, surfactants, and polymers. Then the shell layer, which is chemical difference from material from the core, and the core, which is the central portion of the gold nanoparticle. Well, due to these three layers, these materials got an immense interest for researchers in multidisciplinary fields. Then we have the biological part of the biosensor. Well, first we have for the creation of the biosensor, it is needed a gold nanoparticle that I already explained, an oligo that will be used as a functionalizing material, and HPV 16, 18 DNA, and also FCD9 DNA. The HPV 16 and 18 DNA is not the whole virus genome. It's only the highly conserved L1 section. This L1 section is part of the envelope of the virus. This envelope is left on the body as a residue of the infection of the virus. Well, and the oligonucleotide or oligo are short synthetic DNA or RNA polymers, which can be from 8 to 50 nucleotides. The one that we're using is around 12 base pairs. Well, this oligo modulate gene expression through Watson-Crick base pairing with targeted nucleic acids. This is important because the way that the biosensor works is that this oligo is complementary by Watson-Crick base pairing to the HPV 16 and 18 highly conserved L1 section. So when this DNA of HPV 16 and 18 is on the same site as the oligo, they actually pair through Watson-Crick forces and create a strong bond. Well, for the functionalization that we're going to call functionalization when we add the oligo to the gold nanoparticles. For this is needed to be the oligo is needed to be modified with TOC6 linker. The addition of the thiol group allows the attachment of an oligo to diverse substrates and molecules such as gold nanoparticles. This is possible due to a strongly bind through covalent interactions. And well, for the results, the methodology followed was first the gold nanoparticle synthesis, then the functionalization, and finally the measurements. For the synthesis, we have photo here. We used the Turkevich method using sodium citrate and heating it up for an hour and using a gold chloride solution. Then the functionalization, I already explained a little about the functionalization, is when we add the oligo to the gold nanoparticle. This is done by using, well, due to the strongly bind through covalent interactions, there is not a lot of steps in this functionalization. You need to only add it and heat it up to develop all the DNA in the oligo. And then finally the measurements. Well, we used 100 picograms per milliliter of DNA of HPV 16 and 18, which are the highly conserved L1 section. And the other DNA, FCD9, is actually a gene that it's in our bodies, it's in our genome. And this gene, it was used as a false positive result. When you add this false positive to the gold nanoparticle functionalized, if you get a response, that means that the oligo is not base pairing with the targeted nucleic acids that we want. It's targeting a lot of different DNA sections. And we only want the oligo and the gold nanoparticle biosensor to attach to the HPV 16 and 18. And well, we have here our results. We did a lot of measurements due to the size of the poster. We're only showing two graphics, and also the graphics are shortened to make it more clear the results. We shortened the measurements from, well, we actually measure in a spectrophotometer doing measurements from 200 to 800 nanometers with steps of 1 nanometer. And we shortened this by in showing here only from 450 to 600. And first we have the gold nanoparticles and the oligo and the DNA. These are the three factors or biosensor uses. First, the gold nanoparticles, which is 2 times. This is due to is 2 times the concentrations. It's 2 times the concentration, the initial concentration. Then we have the oligo that we measure from 0.25 to 1 micromolar, and we used 100 picograms per milliliter of DNA, 16, 18, and FCD9. These are our results. Here we have that on the blue squares, we have the gold nanoparticles, only the gold nanoparticles, measured as red as can be seen in the image. And then on the red circles, we have the functionalized gold nanoparticles, which these ones are functionalized with 0.25 micromolar oligo concentration. And then we have HPV 16, HPV 18, and the FCD9 gene. Well, first we can see that the 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 measurements actually differentiate between HPV 16, 18, and FCD9. But these results can be increased when the concentration of the oligo is increased from 0.25 to 1 micrometer. In here, we have that the gold nanoparticle have the same absorbance measurement, and then we have the gold nanoparticle functionalized with 1 micrometer of oligo. Then we have the 16, 18, and FCD9 genes. As can be seen in the graphic, these measurements actually differentiate and create a way to differentiate and to detect the different HPV 16 and 18 and FCD9. Well, as I already explained, the gold nanoparticle, when it's chemically synthesized, has an unknown concentration, but these do not change a lot from batch to batch synthesize because theoretically we are using always the same method and the same solutions. So this unknown but expected concentration is called 1x or 1 times. From this concentration, it is possible to centrifugate, remove the supernatant, and add more gold nanoparticles to increase the number of the gold nanoparticle or increase the solution concentration. That's why we are using 2 times gold nanoparticles. From this and more results obtained, we change the gold nanoparticle concentration, the concentration of the oligo, and the DNA concentration. These are the best results that we obtain, and due to the size of the poster, we are only showing these 2 graphics. But well, from this and more results obtained, it was possible to determine that when the concentration of nanoparticles in solution is bigger, the Plasmon response effect results in a bigger value of absorbance detectable. Also, that with the correct amount of DNA, it is possible to differentiate the type of sample or viral DNA, as can be seen here. If you change the oligo, you get to differentiate the DNAs. The amount of DNA binded to the biosensor changes the absorbance value. From this behavior, it should be possible to create a calibration curve that correlates the amount of DNA and absorbance at the highest wavelength peak. This is something that we are currently working on. We are not only changing the concentration of gold nanoparticle oligo, but we are also changing the DNA concentration of the samples to create a calibration curve that can differentiate and also quantify the amount of DNA and possibly detect how long the infection has been in a person. Well, finally, we conclude that it is possible to detect and differentiate HPV 16 and 18 from a false positive FCD9. In all the measurements, the FCD9 was the lowest absorbance, and the HPV 16 and 18 was on the top, always lower than the functionalized gold nanoparticle due to the increase in the volume and the size of the gold nanoparticle. But well, it is possible to detect and differentiate HPV 16 and 18 from a false positive FCD9 and a functionalized gold nanoparticle when a one-time concentration is used. When using a bigger concentration of nanoparticles, such as 2 times, it is possible to detect and differentiate between samples. Well, for a quantification and detection, the next step, as I explained, should be using a bigger and lower concentration of DNA with 2 times gold nanoparticles and 1 micromolar oligo to create a calibration curve. using the best results that we have with whole nanoparticles and oligo, change the DNA to get a calibration curve. And this is all the poster. And here in the QR code, you can scan it and see or bibliography or results in Excel, and also the contact information of the team. Thank you.